Hello, friends. My best wishes for the World DNA Day today. I'm speaking to you on request of Balaji, whom I met at a recent workshop that we conducted at our center. I was very happy to see his enthusiasm towards research and his interest in learning more than what is routinely taught in MBBS. I was also an MBBS student just like you few years back. However, after my MBBS, I entered into MD in pediatrics. And after that, I tried out doing private practice and working in some hospital. But I was not really happy doing the routine work of seeing patients and prescribing medicines. And that is when I came to know about the DM program in medical genetics at Sanjay Gandhi Postgraduate Institute, Lucknow. So I decided, let's give it a try. Although genetics is really a research subject, but there was slowly some progress happening and clinical utility was coming up. So I joined there for a DM program, and during those 3 years at the institute, I learned a lot about how practice should be done based on evidence, how we can use the principles of genetics into clinical practice, as well as it created an interest in research in me. So after that, I decided to join a research institute and follow my career into research. And I can assure you that after almost 15 years of being in the research field, I'm as happy as I would have been while seeing patients. It is very important for clinicians, for medical graduates in our country to enter into research field either as a full-time career or you can take up research in whatever field that you are working. It's not really necessary that you have to become a scientist to do research. All of you will be graduating, then doing some post-graduation, and you will be reaching some or the other hospital or a medical college. Wherever you go, try to do some sort of a research in whatever facilities are available so that that will help you to build your academic aptitude to help students to come into the research field, and you will surely enjoy it. I hope at least some of you are able to take up research as a career, and we have so many examples of clinicians who have taken a full-time career into research and they are doing very good work. India is really in need of physician scientists, and I hope some of you at least will take up this field. Best wishes for your future, and thank you, Balaji.
Hi all. Hi Sudarshan. I congratulate you on your recent endeavor. You have been a very enterprising person. And of course, now you have understood the need of the hour and you've started a forum on genetics to educate the younger generation and the medical fraternity about the importance of this futuristic field called genetics. So I really congratulate you with all my heart and wish you all the very best in your recent endeavor. So, as per your request, I'm just highlighting my role as a pathologist and what I do in my laboratory on a routine basis. So, as a pathologist, my job revolves around examining tissue samples from patients and identifying if there are any abnormalities or diseases that may be present. So, this involves using basic stuff like microscopes to examine the body fluids, which are called the cytology samples, or the biopsy samples. It could either be a small sampling of the entire disease process, which is called as a throughput biopsy, or the excision of the entire lump or the disease, okay, which is either called as an excision biopsy or a resected surgical specimen. And I do some special stains whenever required, and also higher end techniques these days like what is called as the immunohistochemistry and the molecular diagnostic methodologies like the polymerase chain reaction, and of course the recently like widely available and widely used next generation sequencing and cytogenetics techniques. And fluorescent in situ hybridization is also another technique which also is used for diagnosing certain diseases, which also is being practiced in my laboratory for the appropriate situation. One of the most important aspects of my job is diagnosing cancers, and I'm very passionate about oncopathology. This involves analyzing, as I said before, the cytology specimens, the biopsies, or the resected surgical specimens to determine the type of cancer and to stage the progression of the disease. After the advent of molecular diagnostics, a pathologist's job is no more just a desktop job sitting in an AC room like it used to be commented upon in the earlier days. And it takes a close interaction nowadays for a pathologist to work with the oncologist to develop treatment plan for the patients and to monitor their disease process. In addition to diagnosing disease, I also play a role in research and education. This is another passion. Education is rather another passion, which I consider as a God's gift.
Hi, I am Dr. Meera Purushottam, the Molecular Genetics Lab at NIMHANS, Bangalore. In our lab here, we do testing for many of the movement disorders that is caused by trinucleotide repeat expansions in many genes. We test for the more common ataxias, that is SCA1, 2 and 3. We also test for SCA12, which is rather common in some communities in India. We test for FRDA, which is an autosomal recessive ataxia. Now, though all these are different ataxia, they occur due to mutations in different genes, and therefore if you have a patient who shows symptoms of ataxia, you are never sure whether it is a genetic disorder or whether it is caused by some other physiological or other kinds of problems. And therefore having a genetic diagnosis is often very crucial because it may decide treatment modalities. And secondly, when it is genetic, we know that it is going to get worse. It also means that other members of the family may be at risk, and therefore it's very important to counsel a family before the testing is done and of course at the time of giving the report.
Good morning, everyone. At the outset, I would like to thank Dr. Sudarshanan for giving me this opportunity to address the members of the Cellular and Molecular Genetics and Translational Research Club in Kilpatrick Medical College. He wanted me to just briefly highlight my brief career in brief, as well as also some of the work which we have done within 2 minutes. So I'll try to summarize as much as I can. So I'm an alumni of Madras Medical College, did my MBBS and MD General Medicine from MMC, and then did my DM Medical Oncology from Cancer Institute, Adyar, and then was fortunate to receive the Commonwealth Fellowship, which I then used to do my PhD in Molecular Oncology from Hammersmith Hospital, University of London. And then on my return, I set up the Molecular Oncology Department with the support of Dr. Krishnamurthy and Dr. Shanta. So this department essentially is a translational research department, and we have been recognized as a center of excellence by the Department of Science and Technology. And our main areas of work has been biomarkers for early diagnosis. So we have developed an assay for early detection of cervical cancer. So this technology has been transferred to HLL Lifecare Limited, Trivandrum. We also have identified blood-based biomarkers for early diagnosis of breast cancer, so which is now being transferred to a company. Negotiations are on. The other part of the main work is also on cellular therapy. So we were the pioneers in the country to develop dendritic cell vaccine and for the treatment of cervical cancer. So we developed the technology, then did a phase 1 study, and then subsequently we have just completed the phase 2 study. The analysis is going on. Meanwhile, we've also been funded by ICMR for dendritic cell vaccine efficacy in relapsed ovarian cancer. So that work is again going on. So apart from this, I also have been running the hereditary cancer clinic and doing the mutation analysis for the common hereditary cancers, initially using DHPLC followed by Sanger sequencing, and subsequently we've been using the next generation sequencing technology. And I do the counseling and do the interpretation of the data, and then counsel the patients again once when I get the results. So this is in brief the work which we have done. I'm really happy to hear that you have started this club and there are quite a large number of members. I really appreciate that you have taken on to translational research, which is where as medicos you need to focus on, and I think you have a huge amount of material. Also you can easily do it. The funding is there from different institutions, governmental institutions, private organizations, etc. So that shouldn't be an issue. So I really wish you all the best. Thank you.
Hi, this is Nagarajan. I am a professor in the Department of Chemical Engineering at IIT Madras. I want to talk to you today about some research that we have undertaken in the last few years that is showing some very promising results. This is regarding the formulation of nano-emulsions of spice oils for combating cancer of various types. It is well known in India that plant and spice oils such as cumin oil, celery, cinnamon, and so on have many beneficial properties, including the ability to treat cancer symptoms. However, the challenge has always been to formulate these oils into a suitable form for uptake by human beings. And this effort at making them into nano-emulsions provides a solution for that. These can be orally administered. And we have done studies in our laboratory, primarily led by my postdoctoral scholar, Dr. Joyce Nirmala, and several others in our group, who have shown that these nano-emulsions, once they have been optimized for stability, are quite effective in attacking cancer cells. And there is some lab level data on cancer cell lines, which indicates that these nano-emulsions can be very successfully deployed to fight cancer. And this work so far has been sponsored through the Institute's postdoctoral fellow scheme, as well as the National Postdoctoral Fellow Scheme, and most recently by the Pratiksha Foundation of Chris Gopalakrishnan and Sudha Gopalakrishnan. We are now just embarking on animal trials, which is a very important second stage in the process. And if these are successful, then the next step would be clinical trials, which again would have to be done in various scales. And but we are very optimistic that a few years down the road, we will have these formulations ready for market sales and consumption by cancer patients. And we certainly hope that this will go a long way towards alleviating their suffering. Thank you.
Hi, this is P.N. Rangarajan. I'm a professor of biochemistry at Indian Institute of Science. I'm just giving this very short interview to just tell you about the work that has gone on in our lab, which has some biomedical relevance. Basically, way back in early 2000s, the country was actually importing hepatitis B vaccine from abroad, primarily Ingerix B and Recombivax. So the government of India actually wanted to indigenously manufacture recombinant hepatitis B vaccine. So what we did is we took the gene that codes for the hepatitis B virus surface antigen, put it inside under the yeast promoter, and put it in a yeast called as Pichia pastoris. And you can now see once you clone the gene coding for the hepatitis surface antigen in a yeast expression vector, put inside the yeast cells. Now yeast cells will now start making this vaccine, and we can break open the yeast cells and purify the hepatitis B surface antigen from rest of the proteins. And if we now mix this hepatitis B surface antigen with aluminum hydroxide, it becomes the recombinant hepatitis B vaccine. So this vaccine is now technology has been transferred to companies in India, and there are now 2 monovalent vaccines. One is called as Elovac B and BEVAC. This is made by a company called Biological Limited. This is by a company called Indian Immunologicals Limited. Both are in Hyderabad. And about 10 years later, they combined the hepatitis B with DPT and HIP to bring out a pentavalent vaccine. Indian Immunological marks it as Vexstar 5. Biologically marketed as a COMBE5. So all these vaccines are now currently in the market. But these research were all done about 10 to 15 years back. And we also made a rather unsuccessful attempt to develop a DNA vaccine for rabies. As you know, the rabies vaccine manufacture is a very cumbersome process where you have to infect mammalian cell lines or duck embryo cell lines with rabies virus, multiply the virus in the cell lines, kill the virus, and then give it as a vaccine. So it's a very laborious process. So we thought whether we can develop a DNA vaccine for rabies. So we simply took the gene coding for the rabies virus surface glycoprotein, but instead of putting it in a yeast promoter and putting in yeast cells, we put it under a mammalian promoter and directly inject the plasmid in our body. So that the plasmid now gets into our cells and the viral antigen is synthesized within our own cells and is present to our immune system. Therefore, we can evoke an immune response. This worked very nicely in animal models like mice and monkeys, but we actually want to do a human clinical trial. But unfortunately, we could not get regulatory permissions those days. So somehow the project was abandoned. So it could not proceed beyond preclinical toxicity studies. So this was our contributive development of an indigenous and an innovative vaccine. The indigenous vaccine effort was rather successful. The vaccines are currently in the market, but somehow the innovative effect of effort to develop a DNA vaccine for rabies could not proceed beyond animal studies. So right now our focus is a little bit away from the biomedical research. We are actually using this Pichia pasteur disease to make recombinant proteins, and everybody uses a promoter called as alcohol oxidase, which is a methanol-inducible promoter. That's the promoter we made for making the recombinant hepatitis vaccine. But now in our lab, we have developed a novel expression system wherein you can actually make recombinant proteins using monosodium glutamate, MSG, which is a well-approved food additive. So we have developed certain promoters in this yeast whose transcription can be turned on by adding glutamate or monosodium glutamate. So we're trying to develop a novel yeast expression system where recombinant proteins can be produced by using monosodium glutamate. So as long as you grow the cells in glucose or glycerol, your protein is not made. The moment you change the carbon source from glucose to monosodium glutamate, this promoter is turned on and you can express the protein of interest. So my goal is to see in this next 2 or 3 years, can we develop this novel expression system. We have actually licensed this expression system to a company called as Biogrammatics in the United States. Hopefully they will now start selling these vectors, and I am hopeful that maybe in the next 5 to 10 years people will start buying these vectors and start making some of these proteins of biological importance. Both are biomedical research as well as basic research. Thank you.
சென்னையைச் சேர்ந்த இம்யூஜெனிக்ஸ் பயோசயன்ஸ் என்ற நிறுவனம் ஓமிக்ரான் வைரஸ் பாதிப்பை எளிமையான முறையில் கண்டறியும் நவீன சாதனத்தை கண்டுபிடித்துள்ளது. இந்த உபகரணத்திற்கு கிரிவிட்டா SARS-CoV-2 என்று பெயரிடப்பட்டுள்ளது. ICMR என்னும் இந்திய மருத்துவ ஆராய்ச்சி மையத்தின் அங்கீகாரமும் இந்த கருவிக்கு கிடைத்துள்ளது. தற்போது ஓமிக்ரான் பாதிப்பு இருப்பதை கண்டறிய 5 நாட்கள் வரை ஆகிறது. இந்த புதிய கருவியின் மூலம் 45 நிமிடங்களில் ஓமிக்ரான் பாதிப்பை கண்டறிய முடியும் என்று கூறுகிறார் இந்த நிறுவனத்தின் விஞ்ஞானி நவீன் குமார். கொரோனா, டெல்டா வைரஸ் பாதிப்பு, ஓமிக்ரான் பாதிப்பு ஆகியவற்றையும் இந்த ஒரே சாதனம் மூலம் எளிதாக கண்டறியும் வாய்ப்பு ஏற்பட்டிருக்கிறது என்றும் விஞ்ஞானி நவீன் குமார் தெரிவிக்கிறார்.
அதுதான் லைக் நான் சொன்ன மாதிரி இது இப்போ இருக்கிறது வரைக்கும் ஹோல் ஜீனோம் சீக்வன்ஸ் பண்ணி தான் ஒமிக்ரானை கண்டுபிடிக்கிறாங்க. இப்போ நாங்கள் கண்டுபிடிச்சிருக்க இந்த புதுசான ஒரு கிட்டு ஒரு புதுமையான ஒரு கிட்டு. இது என்ன பண்ணும் அப்படின்னு பார்த்தீங்கன்னா, ஒரு நபருக்கு கொரோனா இருக்கா இல்லையா, அது டெல்டாவாக இருக்குமா இல்லை ஒமிக்ரானாக இருக்குமா, ஸோ இந்த ஒரு 3 விஷயத்தை அது உங்களுக்கு ஈஸியாக எளிமையாக கொடுத்துரும். அதுக்கேற்ற மாதிரி டாக்டர்ஸ் ட்ரீட்மெண்ட் பண்ணலாம். இப்போ பார்த்தீங்கன்னா இப்போ ஒமிக்ரானை எப்படி கண்டுபிடிக்கிறாங்கன்னு பார்த்தீங்கன்னா, ஒரு பேஷண்ட் கிட்டேருந்து எடுத்து மரபணு சோதனை பண்ணுறாங்க. அது யூஸ்வலாக ஒரு 6000லேருந்து 10,000 வரைக்கும் ஆகும். பட் இப்போ நம்ம பண்ணியிருக்க இந்த புது கிட் பார்த்தீங்க அப்படின்னா இப்போ ரொட்டீனாக எப்படி ஆர்டிபிசிஆர் பண்ணிட்டு இருக்காங்களோ அதே வேலையிலோ இல்லை கவர்மெண்ட் ஃபிக்ஸ் பண்ணக்கூடிய ஒரு 1000 ரூபாயிலேருந்து 1500 அந்த ரேட்குள்ளேயே உங்களால் பண்ண முடியும். 45 நிமிஷத்துக்குள்ளே பண்ண முடியும். அது 7 நாள்லேருந்து 45 நிமிஷம் கூடியன்றது ஒரு பெரிய அட்வான்டேஜ். இதை பேஸ் பண்ணி என்ன அட்வான்டேஜ் அப்படின்னு பார்த்தீங்க அப்படின்னா, ஒரு பேஷண்ட் ஒமிக்ரான் இருக்க பேஷண்ட்டோட ஸ்ப்ரெட் வந்து ரொம்ப அதிகமாக கம்பேர்ட் டு மற்ற வைரஸஸ். அப்போ வந்து நீங்கள் அவங்கள தனிமைப்படுத்தி ரொம்ப எளிமையாக தனிமைப்படுத்திடலாம். இப்போ ஏர்போர்ட்லேருந்து வரவங்கள சீக்கிரம் ஸ்க்ரீன் பண்ணி நம்ம உடனே பண்ணிடலாம். ஸோ அப்போ வந்து உங்களுக்கு வந்து இந்த சமுதாய தொற்று அப்படின்றத நீங்கள் அவாய்ட் பண்ண முடியும். அட் த சேம் டைம் இப்போ நீங்கள் ஒரு டாக்டர் வந்து ஒரு பேஷண்ட் ட்ரீட் பண்ணுறாரு அப்படின்னு பார்த்தீங்கன்னா, டெல்டாக்கும் ஒமிக்ரானுக்கும் ட்ரீட்மெண்ட் வேரி ஆகும். ஏன்னா டெல்டா வந்து கொஞ்சம் சிவியராக இருக்கிறதுனால அதுக்கேற்ற மாதிரி சில மோனோக்ளோனல் ஆன்டிபாடிஸ்லாம் யூஸ் பண்ணுவாங்க, ஆன்டிபாடிஸ் யூஸ் பண்ணுவாங்க. அது வந்து கொஞ்சம் காசு அதிகமாக இருக்கும். ஒரு 60,000லேருந்து 70,000 வரைக்கும் இருக்கும். வேற இந்த ஒமிக்ரானுக்கு பார்த்தீங்க அப்படின்னா உங்களுக்கு அதோடைய சிவியாரிட்டியே ரொம்ப கம்மியாக தான் இருக்குது. ஸோ ஒரு 3 நாள் 4 நாளுக்குள்ளே உங்களுக்கு அது இதுவாகிட்டதுனால நீங்கள் இந்த பேஷண்ட்டை ட்ரீட் பண்ணுற விதமும் அது வந்து ரொம்ப ஹெல்ப்ஃபுல்லாக இருக்கும் இந்த விஷயத்தில்.
This is the animation of hand movements that is in real time and modeled in MATLAB Simulink. In order to do this, quaternion data from all sensors are read through the serial communication in MATLAB Simulink. The relative quaternion at each joint is computed using the conjugate quaternion operation, which is later converted to Euler angles and then animated. But please note, this is not the only way to do this animation. It is not always necessary to convert the quaternions into Euler angles. There are some softwares and tools which take in quaternions directly and animate in real time. We will also demonstrate that in a few seconds.
This is a VR based application which is developed using the Unity game engine software. This application accepts kinematic data from 2 HKMS devices and uses it to animate a pair of virtual hands in VR space. The animation of the virtual hands is done using raw quaternion data.
This is Lily from the Mouse Genome Engineering Facility. So MGF is a national facility that was born at the end of 2014 thanks to DBT. We cater different types of services, including mouse sperm or embryo cryopreservation. We can cryo-recover these stocks and offer services to rederive different mouse lines as well. And thanks to the nice setup that we have here with ACRC SPF Mouse Facility, we are now one of the largest Indian mouse repositories, maintaining over 360 different transgenic mouse lines. We also offer services to make new mouse models for your research purposes using CRISPR-Cas or other gene editing tools. We also have been very active in organizing training and workshops to allow other scientists to acquire these technologies and other facilities to act as regional hubs across India to enhance the sharing of resources among Indian scientists. Have a wonderful National Science Day and hope to see you soon here at BLISC. Bye-bye.
Hi, I am Arindam Maitra from the DBT National Institute of Biomedical Genomics, Kalyani. One of the research areas that our lab works on is the genomic and epigenomic underpinnings of preterm birth. We are using the Garbhini cohort located in northern India, which is a large prospective pregnancy cohort. And we have some recent findings which are novel in nature and reported for the first time from South Asia and globally.
Hello everybody, greetings to the MBBS Students Club at Kilpack Medical College. I am happy to be a part of this. I am working in Jipur, Puducherry in the Department of Pharmacology. My area of interest is in tuberculosis. So I have been working in research related to tuberculosis, like adverse drug reactions, drug resistance in tuberculosis. So I'll be very happy if few of you could take up this area, because this is an important requirement for our country, wherein tuberculosis research is still in need. So all the best for all the academicians, clinicians and researchers of future. All the very best. Thank you.
Hello everyone, I am Dr. Nidhi Puranik, presently working as Research Assistant Professor in Yongnam University, Daegu, South Korea. My research work is development of therapeutics molecules for neurodegenerative and neuropsychiatric disease. Presently, I am just focusing on the development of phytochemicals based therapeutics molecule for Alzheimer and autism disease. I just would like to tell you, please show much more interest in research in medical field, because we need a lots of medicines, vaccine and the drug for various different diseases. So please continue your research, show as much as interest in research, and if you need any kind of help regarding my research work, please let me know. I am always available for you guys. Thank you so much. Bye.
Hi everyone, I am Dr. Beryl Vedha, working as scientist in Satyabama Institute of Science and Technology. My area of interest is neural stem cell regeneration. So I am mainly focusing on neurodegenerative disorders, particularly Parkinson's disease. The model animal I am handling is zebrafish and mice. So we all know about turmeric, right? It do wonders. So it has an important chemical compound called aromatic turmeron. It has the ability to regenerate neural stem cells, but the molecular mechanism is still unknown. So we are mainly focusing on the molecular mechanism, how this aromatic turmeron regenerate the neural stem cells. So this finding will definitely make a breakthrough in the field of neuroregenerative medicine. So thank you for this opportunity. Wish you all a very good success.
Hello, I am Dr. S. Johnson Retnaraj Samuel from Satyabama Institute of Science and Technology. So my area of research is regeneration and stem cell research, and I am using earthworm and zebrafish as animal model system for my research purpose. So far we have developed an FBS alternative. So everyone knows about the FBS, fetal bovine serum, which is very much needed for animal cell culture. So due to ethical concerns and lot of issues, so it is very difficult to get the FBS, fetal bovine serum. So we have a very good alternative for FBS, isolated from the earthworm. So this our alternative of FBS, which is isolated from the earthworm. So as mimics very well in cell culture. So far we have developed the cell culture medium, serum-free cell culture medium, and currently it is in the patent stage. Right, this is one of the research, and the second we have isolated some of the fluoropores chemicals which exhibit the fluorescence. So from the earthworm, so it has a dual role. So it has antibacterial activity as well as it has the wound healing capability. So and this will help in many applications. Like we can use it for even human also. So this is our research, what we are doing in Satyabama Institute of Science and Technology. Thank you.
At the outset, I would like to thank Dr. Sudarshan Balaji and the committee members for their efforts in establishing a clinical, cellular and molecular genetics and translational research club at the Government Kilpack Medical College and Hospital at Chennai. I understand that the overarching objective of the club is to inculcate training in basic and advanced genetics to students pursuing MBBS from various medical institutions in the state of Tamil Nadu. Friends, As we are celebrating World DNA Day on 25th April, it is befitting to remember the immense contributions of the medical and scientific fraternity in the area of cell and molecular biology, immunology, which have not only helped us to understand, to solve the clinical riddles of inherited diseases, but which also have helped immensely towards understanding the very basics of gene-environmental interactions. Through these discoveries only, we have now realized the big picture of the developmental origin of adult diseases. In today's time, a well-trained clinical genre of scientists is highly warranted when lifestyle diseases are contributing enough towards overall mortality and morbidity. More so for a country such as ours, which suffers from economic and environmental perils. The burden of the disease is alarming in our country, as you all know. We are now the most population nation crossing China. Therefore, strengthened healthcare infrastructure and ecosystem is the need of the hour. I am privileged to be associated with the various academic programs of the great college, that is the Kilpauk Medical College and Hospital. And basically, I was fortunate to have visited your great institution last year for a flow cytometric training event as well. We at ICMR near Bhopal are engaged in understanding the ramifications of mitochondrial-induced epigenetic modifications in specific cohorts exposed to various environmental pollutants, including airborne ultrafine particulate matter. We are also making concerted efforts towards understanding the role of nanobiosensors and trying to develop and validate various point-of-care approaches for the early identification of environmental-associated non-communicable diseases. Friends, we extend you warm greetings and wish you all the best for the future academic endeavors of the club. May Almighty bless you all. Vanakkam and Namaskar. Thank you.
Good day to all the aspiring doctors. I'm Dr. Bhavani, Senior Scientist and Genetic Counselor. I'm very happy and feeling privileged to be a part of this World DNA Day celebrations. This day is celebrated globally to mark the recent advancements in genomics and to explore its impacts in lives. My routine works includes developing molecular diagnostic kits, analyzing and reporting cases of molecular and cytogenetics. And as a genetic counselor, I'll be interacting and counseling patients and their parents. On a daily basis, we come across many children and neonates with congenital genetic defects, most of which can be managed when identified and treated at a very early stage. As in case of ambiguous genitalia, where an early diagnosis is crucial for proper development of their physical and mental health. The recent advancement has given us many molecular tools, which is for accurate diagnosis of the disease, but each has its own advantage and limitations. So a detailed analysis of the case by both the clinician and genetist is crucial in selecting the appropriate test for the accurate diagnosis and a better patient care. As a researcher, we love problems, and as a doctors, you need solutions. So, the collaboration of a physician and a dentist is the key that is bridging the gap between the research and a practice. I hope these club and the meetings that you conduct will help you to get motivated for your future career and to take your research as a career. Thank you all. I wish you all a very bright future. Good day.
Warm greetings to all of you. It is really a pleasure addressing this gathering of young budding scientists. From time immemorial, man has advanced his knowledge of disease. From the hit and trial methods of a few centuries ago, we have evolved to the present state of understanding, which includes not only general pathology of disease, but its molecular and now even genetic basis. In fact, today, epigenetic basis is seen to be as important as the genetic and molecular bases. Genetics and epigenetics have opened a plethora of possibilities, and especially in research, so much so that management and sometimes even prevention are based on the current knowledge in these fields. And that is what personalized medicine is all about. How to decide the type of medication and its dose depending upon the patient's individual pharmacokinetics and genes. And as we speak, this knowledge is increasing in leaps and bounds. Yet, there is so much more to discover. So many uncharted areas of basic mechanisms that are being uncovered, and we can all work together or individually for a very long time. What better example of this can there be, can any country actually have, than our very own C. Radhakrishna Rao, a 102-year-old statistician who was a recipient of the Padma Bhushan at the age of 47 years and the Padma Vibhushan at the age of 80 years, and now has been awarded the Nobel Prize for his exemplary contributions to statistics. And as our scriptures say that our success is measured by our effort and our intent, I wish all of you a great life of extended research, world-class research, especially in the field of genetics, to commemorate the theme of today, World DNA Day. Thank you.
Hi, National DNA Day wishes to you all. This is of course being celebrated in US by NHGRI, National Institute of Health, but all over India this is also being celebrated, and so is India. And all over the world it is being celebrated, and so is India as well. I'm Prashanth N Suravajhala. I'm a principal scientist at America School of Biotechnology under Mishra in Kerala, India. We work on systems genomics of rare diseases in CFO state, and we try to identify the known unknown regions of the genome and infer the causal or pathogenic variants attributed to various diseases. So, as we talk about DNA has got 4 bases, 3 bases to make up one triplet that makes up one codon. And so I talk about 3 important C's that one can make up of its scientific career. One is consistency, second one is continuity, and the third one is credibility. Likewise, I'll also talk about 3 E's. One is enthusiasm that one should have. E is not to keep expectations in life, those serendipitous with the research. And third and most important thing is keep in view of the epigenetic spectrum, which means the environment plays a very important role on how we are influencing. So that's what this DNA is all about. Happy National Day. Thank you.
Hi Balaji, I am Dr. Usha Dutta from CDM, Hyderabad, and I am a cytogeneticist. I am really happy, Balaji, that you have initiated this society, creating awareness in genetics to the medical students. At CDMT, I'm involved in services as well as in research. Services we provide in cytogenetics, molecular cytogenetics and molecular biology. And molecular cytogenetics we include the techniques like fluorescence in situ hybridization, FISH as an MLPA, multiplex ligation dependent probe amplification, and microarray data analysis, and exome sequencing, and also whole genome sequencing. Whole genome sequencing is at the research level. My research interests are in long read sequencing of the copy neutral variants like balanced reciprocal translocations and balanced inversions, and also then I'm interested in development of the novel diagnostic technologies. So any one of you are interested in learning these techniques or knowing about this, any queries you have, you can contact me. My
Email ID is usha@cdfd.org.in. And last year I have written a book titled Essentials of Cytogenetics and Molecular Cytogenetics Laboratory Testing, which is published by Cambridge Scholars Publishers. So any students who are interested in going through the techniques can go through it, because it is a quick reference, as well as the people who are wanting to establish the techniques in the labs also can go through that, because it is all about establishing the techniques at the lab level also. With that, I wish you good luck for your society. Thank you very much.
Hello everyone, and my dear students. Myself is Dr. Jaiswal. I'm a professor and director in Blood Center, Transplant Immunology and Flow Cytometry Lab at Mahatma Gandhi Medical College, Jaipur. First, I would like to extend my thanks to CCMGTR Club of Government Kalpak Medical College, Chennai, for giving me this opportunity on this very special occasion day of DNA, that is a DNA Day, which is observing on 25th of April every year, on the occasion of the completion of Human Genome Project in 2003, as well as the discovery of the double helix of DNA in 1953. Just I wanted to say, I started my career way back in 2003 in the pathology, and at that time we started our career with seeing only morphology. Most of the molecular kind of things are our dream at that time. But as the time passes, we we initiated the concept of morphology to molecular at our center, and at presently we are taking care of everything from morphology to molecular under one roof, and it is possible because of the great support of our Honorable Emeritus Chairperson, sir, and presently we are taking care of all kind of transplant at our center, including solid organ and bone marrow transplant, and as well as we are taking care of all the molecular testing, especially for monitoring of organ transplant program by Luminex. We are doing HLA typing, which is a DNA based test. We are doing flow cytometry for leukemia and lymphoma. We are taking care of next generation sequencing for the high resolution typing, as well as the profiling of various neoplasm, and as well as we are also taking care of karyotyping and cytogenetics at our center. Just on this special occasion, I extended my good wishes to all the young buddies who are still in the MBBS phase, who are studying. There's a lot of things when you come out after the MBBS phase or your MBBS course, because we are living in the era of molecular testing. So there's a lot of scopes for the molecular testing, especially DNA-based testing for the sequencing, and my good wishes to each and everyone. Thank you very much.
Hello everyone, I'm Michael Gromiha, leading Protein Bioinformatics Lab in the Department of Biotechnology, IT Madras. Proteins perform various functions in living organisms, and if there is a mutation, for example here T2D, may alter the structure of the protein and stability as well as function, and some of them may lead to diseases. It is very important to understand the effect of mutations at the sequence level and at the structure level, as well as at the functional or disease level. So our lab is focusing on understanding the effect of these mutations and developing computational tools for identifying the specific disease-causing mutations. Specifically, we developed a method for identifying cancer-causing mutations in glioblastoma, the GBM driver. It takes the mutation information and calculates the features from the sequence and structure, and it predicts the effect of these specific mutations, whether it is a driver mutation or a parasite mutations. Another example is ALS disk. Here we try to identify the Alzheimer's disease causing mutations using sequence and structure information, and it takes the sequence here and their mutations, and it predicts the effect of the mutations as either disease causing or neutral. We have developed various computational algorithms for understanding these mutations, and which are available in our website. And you can see my books. This will also help to get more information regarding the effect of these mutations and disease causing mutations. These are my lab members, and thanks for your kind attention. My best wishes for your bright future in your career.
Dear students, I understand that you have started a clinical cellular and molecular genetics and translational research lab some time ago. This is a great step to create awareness among clinicians on clinical genetics, human genomics, and also various genetic and molecular diagnostic tools. Looking 60 years back, when I entered the MBBS course, the then available biochemistry textbook described that the function of DNA is unknown and the basis of heredity resides in proteins. According to that textbook, the protein biosynthesis is taking place by replicating the available protein. We have traveled a long distance from that past. Molecular biology is defined as the study of gene structure and function at the molecular level. Molecular genetics is the scientific discipline concerned with the structure and function of genes at the molecular level. It is estimated that more than 6% of all infants born alive suffer from genetic diseases and 1% from chromosomal aberrations. These involve alterations in DNA makeup. To take an example, congenital sensory neural hearing loss is the most common sensory disorder, with approximately 1 in every 1,000 newborns. Most of them are due to genetic causes, and there are more than 90 causative genes identified. This reflects the complex clinical and genetic landscapes of hereditary hearing loss. Gene therapy was once considered fantasy. However, thousands of individuals have already undergone human clinical trials. Gene therapy is effective in inherited disorders caused by single genes. Most dazzling accomplishments are severe combined immunodeficiency, Duchenne muscular dystrophy, cystic fibrosis, hemophilia, spinal muscular atrophy, and so on. CRISPR technologies have revolutionized the genetic engineering. With CRISPR, it is now possible to modify, delete, insert, activate, or inactivate any gene as well as fine-tune them. Thus, a troublesome gene can be inactivated. CRISPR technology is often hailed today as the future therapy of genetic diseases. Cell and molecular diagnosis, that is CMD, have become a separate entity in medical laboratory setups and have become an important tool for better diagnosis and treatment. CMD gives insights into the pathogenesis and prognosis of various diseases.
Hi, I am Professor Sujatha from IIT Madras. Today I would like to introduce a new concept of optical tissue biopsy to you. It is essentially a diagnostic technique based on light-tissue interaction. You all know what is conventional tissue biopsy is. You will extract the tissue and subject it to under a lot of mechanical and chemical processes. And then finally, the pathologist will look under the microscope to see the condition of the tissue to make a diagnosis. It is an invasive, risky, costly, and time-consuming process. You know about it. Now, what we propose through optical biopsy is an alternative method of doing the analysis for diagnosis. And what advantages it can offer is it's a non-invasive, in vivo, near real time, and it uses only portable instrumentation. Absolutely no tissue processing involved because we are proposing it as an in vivo technique, and we can have additional information such as tissue perfusion when we adopt optical biopsy. The entire phenomenon is based on light tissue interaction. So in optical biopsy, we consider the tissue as an optical material with embedded scatterers, absorbers, and fluorophores. And depending on how light is interacting with these constituents, it will give different responses. Now, different modalities of optical tissue interactions are existing, and you can get the biological information such as cell density, size, orientation, the tissue perfusion, the quantitative information about the tissue constituents in a completely non-invasive environment. So that's what our research is. Some of the ongoing projects in our lab are listed here, and 2 minutes are not sufficient to explain everything. So I would request you, whoever is interested, to visit our lab or visit at least our website to know more about our research. Okay, thank you.
Namaskar. Best wishes to Sudarshan Balaji and all the club members of your clinical cellular molecular genetics club, and also congratulate your teachers who are supporting you in these activities. With the advancements of genetics and molecular biology, lot of things have become clinically applicable, and the whole medicine now is centered around DNA molecule. At this point of time, it is very important that our medical education has lot of these information in their training during MBBS and postgraduate courses. Your club is very pertinent at this point of time, and I am sure will play a very important role in making students and the faculty aware about the advancements in molecular medicine and make them prepared for the practice of medicine in this current era and the coming era. With availability of DNA-based technologies, a lot is possible now for the patients, say prenatal diagnosis, pre-symptomatic diagnosis, and gene therapy. This has evolved many issues related to ethics as well. All of us are carrying DNA and some variations in it. So medical genetics is the specialty, I think, which has the varied ethics related issues and in much more numbers as compared to any medical specialty. And hence it is very important that as technology advances and many things are possible, before we implement them in our clinical practice, it is important that we see which of these investigations are clinically meaningful and can be used for the patients. So it is time that in addition to our technology, we are also aware about its various implications for the society and learn about it. And for this, the genetic literacy amongst doctors and laypersons is very, very important. I am sure your club will play these varied roles in your teaching, training, as well as making.
Hello. It's nice to know about your initiative of knowing about the power of research in medical situations. My name is G.K. Suresh Kumar, and I'm going to talk about reactive species in cancer treatment and management, some of our work. Reactive species are molecules such as superoxide, hydroxyl, peroxide, etc. And they play very important roles in cancer homeostasis. They are very important molecules, but they can be deleterious at high levels intracellularly. In cancer, they play diametrically opposite roles. At high levels, beyond homeostatic levels, they can turn normal cells to cancerous ones. But at much higher levels, they are actually used to kill cancer cells. In fact, a reactive species-based killing is the mechanism by which most chemotherapeutic drugs work, as well as radiotherapy works. The basis is something like this. The basal reactive species, level reactive oxygen species especially, is higher in a cancer cell. And therefore, if you increase that level through drugs, then you enter into a regime of death more in the case of cancer cells compared to normal cells. There are many agents that induce reactive oxygen species production in the cell, pollutants, virus, tobacco, smoke, and so on and so forth, as well as they are caused by intracellular in intercellular events. These are normally caused due to various cell mechanisms, and they could be abnormal at times. What we discovered was that there are time-based rhythms of reactive oxygen species, for example, superoxide. This we found in a particular colon cancer cell line, HCT116, but we expect it to be a general enough phenomenon. We found that there is an endogenous rhythm of superoxide in cancer cells. And when you can do an analysis and find out that the time period of this rhythm is about 24 hours. What we also interestingly found was that a reactive oxygen species generator, menadione, which is some sort of a, let's say, a drug, can cause changes in the rhythm characteristics. There is a certain way it causes the change which we found. So the menadione or a drug treatment causes a change in the rhythm characteristics of a reactive oxygen species, which at higher levels kills cancer cells. And what we found was that if you can match the rhythm of these, if you can match the rhythm of these reactive species to the time of administration of the drug, then the killing could be much higher. For example, the cytotoxicity of curcumin, which is a known cancer drug, when added at 4 hours from the basal stage instead of at 0 hours and so on and so forth, was 27% higher. The cell killing was 27% higher than when added at 0 hours. It's one of the results. Which means that the drug can be at least 27% more effective if added at the appropriate time in the intercellular reactive species rhythm. And therefore, by a mere change in the time of administration, the same drug at the same concentration can be much more effective, which can lead to lesser side effects. If you need further information, please contact me. Wish you a happy DNA day. Bye.
Hello everyone, myself Sabrinathan. I'm a faculty at NCBS. It's such a great pleasure meeting you all on this wonderful DNA Day celebration. Thank you, Mr. Sudarshanan, for this opportunity. So, since the discovery of DNA double helix structure, tremendous technological and molecular biology advancements have been made to understand the function of DNA in health and diseases. One such kind is DNA sequencing, and I'm sure that you are all familiar with this, because in our lifetime, we all witnessed the application of DNA sequencing in the real-time monitoring of COVID viral genome and sequencing. So this kind of sequencing approach is not specific to virus or pandemic. It is also important for all other diseases that are related to DNA. For example, in our lab, we are interested in understanding cancer evolution, and we do this by looking at the DNA of patient tumors. In particular, we are interested in understanding how somatic mutations accumulate in the tumor tissues and which mutations are involved in the cancer progression, and whether we can use this information to identify treatments that are suitable for that patient. In relation to that, in recent years, our lab has been focused on finding biomarkers so that we can predict which mutations or which genomic alterations in a patient tumor is suitable for treatments such as immunotherapy, which is a recent paradigm in the cancer treatment. With your medical background and your bright young mind, I really encourage everyone to get familiar with this kind of advanced sequencing approaches and applications, so that in your advanced study you can use these approaches to study the health and diseases that you are interested in, and this will tremendously benefit both the basic biology and the translational aspect. With this, I would like to thank you all for this opportunity, and I am happy to connect with any of you if you like to learn more about the genome sequencing in the cancer study. With this, I would like to wish you all a very best in your studies, and have a wonderful day. Thank you.
Hello everyone. Firstly, I would like to congratulate the organizers for this wonderful initiative and excited to communicate with you all on this platform. Our lab is working on developing lipid nanoparticle-enabled nucleic acid therapeutics for inherited diseases like hemophilia, where we developed liver-targeted lipid nanoparticle system to deliver functional factor 8 or factor 9 to restore coagulation cascades in hemophilia A and B respectively, where we completed our preclinical studies. And in our other project, we developed mRNA-based vaccine for SARS-CoV-2, where spike encoding mRNA is delivered with a targeted lipid nanoparticle system that specifically targets dendritic cells in vivo for enhanced T-cell responses. Here, our efforts on developing lipid nanoparticles to deliver nucleic acids into particular cell types. Unlike other drugs, nucleic acids cannot be delivered alone, they need vector. Here, the viral vector or non-viral vectors. Viral vectors are successful in delivering pDNA, but they are pretty expensive. The best example is Zolgensma for DMD, which costs over 16 crore rupees in India. Here in our lab, we are making attempts to develop an affordable, economical nucleic acid therapeutics. Thank you.
Dear friends and colleagues, I am Dr. Indumati, Head Department of Medical Genetics, Apollo Main Hospitals, Chennai. Best wishes to each one of us and to Clinical Cellular Molecular Genetics and Translational Research Club, Government Kilpak Medical College, Chennai, on this DNA Day. This is a moment of pride being a geneticist, as DNA is in spotlight, and glad that many of us are exploring genomics for better patient care and management. Human Genome Project and next generation sequencing technology, especially the drastic drop in the sequencing price, is leading us to move forward with the genomic medicine. In our country, there is a huge burden for genetic diseases. It is not only because we are most populated, but we are also highly heterogeneous with inbreeding practices, consanguineous marriages. This leads to the accumulation of the deleterious variants and makes us prone for autosomal recessive conditions. In addition, the present lifestyle pattern, in addition to the genetic predisposition, makes us prone for several multifactorial disorders like diabetes mellitus, hypertension, cancers, and so on. Nevertheless, we are also seeing more significant de novo variants, the sporadic variants in our routine practice. Genomics thus helps in the diagnosis, prognosis, management of patient by providing appropriate tests with more diagnostic yield and a tailored and a targeted therapy with respect to the variants. And I want to emphasize here there is so much importance for having a regional databases. For example, in our practice and with the with the evidence of lot of consanguinity in our South Indian population, we see lot of Fanconi cases. So I would like to appreciate here that there is so much important that we have lot of idea about what is happening in our current clinic, in our current population, in our current area of prevalence of genetic disorders. So uh, so there is lot of hope now coming up with respect to treatment and handling of genetic disorders. One is that the National Rare Disease Policy 2021, wherein there is a support for management of conditions, one-time management, and for certain follow-up conditions can be very well handled here through this disease policy. And there is a registry for rare diseases by ICMR, which is helping us to establish the prevalence and also establish the protocols for management of our patients with respect to our own data. And we have a Guardian program from Delhi, which helps us to do a diagnosis using the genetic methodologies. And it is so evident that we have to establish patient support groups, because this will lead us for more connection and support, the especially the psychological support. Especially this is not only this specific patient is having the genetic disease, but there are a lot more there already who will help them to go through the process in a smooth and a better way with much with knowledge transfer. And as we have discussed earlier, establishing the disease specific registry is also important, but these are very much doable with very limited resources in terms of manpower as well as funding. So I wish this presentation is helpful, and wish you all once again all the success for your society, and also wish you all the best. And this is my team. I would like to appreciate for all the group work that we do here. Thank you.
Hi, I am Saramagavan. I am a scientist at Center for Stem Cell Research, which is a unit of INSTEM Bangalore, and we are here at Christian Medical College campus in Vellore. My lab develops gene-edited hematopoietic stem cells for the gene therapy. So our working model is to collect the patient hematopoietic stem cells. We know the underlying genetic cause, so we use gene editing reagents to correct the genetic mutation, and we will transplant the modified stem cells back into the patient. So because we have corrected the stem cells and we have corrected the DNA, we are expecting to reverse the disease. So right now we are focusing on beta hemoglobin disorders such as sickle cell disease and beta thalassemia, and we are also working on infectious disease like HIV. We are expanding the scope of the disease correction, and if you are interested in our work, please contact me. Thank you.
Hello, my dear students. You know that 25th April is being celebrated as the DNA Day, and we start in 1953 when the first DNA report was published, and 50 years later in 2003 the first human genome map was published. And the next 20 years there was much more technological advances leading to our better understanding of the human genome, and still we don't know about the exact sequencing of the human genome. They are based on the bits and pieces woven together to build up the human genome map. And in 2022, one of the advances was the gapless human genome sequencing. Still, there are other regions in the genome of an individual which are repeated sequence. Now these repeated sequences provide diversity to the human population. And that's why when we talk about the human genome, one thing is that there is a one humanity but multiple genomes. And as a part of the career in the medical genetics, we have to understand that the God has always created something hidden to us, and we have to learn this a long way in the coming years. That is, what is the written things in the genes which can dictate our future. And that's why for interest in the medical genetics is very important, as we know that it is now having the impact on all the biology. I wish you all success for studying the genetics as a unique subject, because it gives a much more curiosity. It elicits your curiosity to the utmost.
Hey, hi everyone. I'm really happy to be doing this video on for the World DNA Day celebrations. And it's really a pleasure to talk to MBBS students for the future of medical treatment in this country. I think I just wanted to say a few words about what is it that we've been doing in our lab. So my lab works on computational approaches to understand and manipulate biological networks. We also do quite a bit of analysis of large biological data sets, typically which we call multi-omic data sets, wherein we have genomic information, transcriptomic information, network information, and so on. And we solve a spectrum of problems. We tackle a spectrum of problems in this space. Our primary focus is on microbiome systems biology, but I think for the MBBS students, what would be more interesting is our work on cancer biology, where we've been applying a variety of machine learning methodologies to try and understand what are the key genes that drive the progression of cancer in various patients. So we've done this on pan-cancer models, meaning across cancers, what are the key genes, which are called driver genes. So these are genes that specifically confer a growth advantage to the cancer cells. So these could be tumor suppressor genes or oncogenes. I'll not get into all the details. You can read more about it in our work. But there are various kinds of genes. But what is very interesting is that cancer is so heterogeneous, right? So the same gene in one patient or the same mutation in one patient may not drive cancer the same way as it does in another patient. So for instance, a particular gene could be a tumor suppressor in a particular mutation in a gene could be a tumor suppressor in one patient, but the same could be something that causes tumor in another patient and so on. So there are these very diverse etiologies for different cancers or even the same cancer across different patients. And there is so much data that's available now from genome sequencing, from doing RNA-seq to analyze the transcriptome or the various RNA transcripts in every cell. And there are also studies looking at proteomics and so on. And the interactions between various molecules in different cells. So can we use all of this information to learn what could be the most important aspect or the most important mutations that drive cancer in different patients? So this is something that we do, and you can check out our lab website at IIT Madras, or also follow me on Twitter, and our Center for Integrative Biology and Systems Medicine, or our Center for Data Science and AI at IIT Madras, where you can read a little bit more about our work on tackling various interesting problems, including a very recent pipeline that we have designed for a GUI pipeline, where you can just put in sequence data and study a little bit more about what are the kind of mutations that are there in the patient and things like that. Thank you, and I wish you all the best and all success.
So, as I told you, they behave as oncogenes or tumor suppressor genes. How they can behave as oncogenes? If the target is a tumor suppressor gene, then the microRNA behaves as an oncogene, because if you suppress a tumor suppressor, then you are becoming oncogenic. So that is the idea. Similarly, if the target is an oncogene, then the microRNA behaves as a tumor suppressor. So in the opposite way, you have to imagine. So for the oncogenic microRNAs are sometimes called oncomirs. The tumor suppressor microRNAs are called tumor suppressor microRNAs. So that is how they were. Sometimes anti-oncomirs also they are called, but don't get confused. Just remember tumor suppressor microRNAs. So for the oncogenic oncomirs, the target is tumor suppressor. For the tumor suppressor microRNAs, the target is oncogenes. So this is what you have to remember. So their expression is controlled like any other gene. So you may have some drawbacks in their expression. Like if there is a mutation in the gene that produced it, then a microRNA may not be produced. So the microRNA biosynthesis is controlled by the same genetic mutations or alterations that happens to any normal gene, like either mutations, deletions, or even the single nucleotide polymorphism. Any defect that happens in the gene can happen to these microRNA genes also, and their expression can be controlled. So this is what about their biogenesis. Then we will pass on to the other details.
Hello, really happy to be talking to all of you through this video. Thank you to Dr. Sudarshan Balaji for taking this video and having me talk about some of the research that my laboratory does here at the Indian Institute of Science, Department of Bioengineering. What we are primarily interested in is to look at how to modulate immune responses using drug delivery systems, biomaterials, maybe develop better vaccines or develop better ways to modulate immune responses for a variety of diseases. And we have a clinical focus in our research. So we do collaborate with many clinicians, do clinical studies. In fact, one of our most forward-looking ideas is to figure out whether we can help people with diabetic foot ulcers. A large proportion of diabetic individuals, so somewhere between 10 to 30%, end up getting these foot ulcers. And many of them, with the current state-of-the-art treatment technology, still do not heal their ulcers. And so through research in our own laboratory and through clinical studies in collaboration with clinicians here in the Karnataka Institute of Endocrinology Research, we have determined that there are certain immune markers that seem to be upregulated in individuals that do not heal their ulcers. And so we have been designing new bandages that can be potentially immunomodulatory in some way and help heal those ulcers faster. So part of it is clinical research where we identify these biomarkers. Part of it is laboratory benchwork research, making these bandages. And we are also doing preclinical research where we test some of these bandages in animal models of research before we can take them forward for clinical tests and trials. Similarly, through some of those studies and through other interactions, we have started networking with clinicians on chronic kidney disease. We've also been working with individuals who work on gliomas, clinicians who treat gliomas, and trying to understand how immune cells are altered both in the brain tumor microenvironment as well as in chronic kidney disease in circulation, and those individuals who undergo dialysis. And the hope is that all of this research will lead to a better understanding of how immune cells change in these individuals, and whether specific treatment strategies, maybe existing drugs but not used for those sort of indications, or newer therapeutic modalities can be identified to promote health and well-being of individuals with chronic morbidity. If you have any questions, I'm happy to answer them. You can write to me. My email address is siddharth@iisc.ac.in. You could look it up on any of my department websites. And wishing you all the very best, and looking forward to seeing many of you becoming more interested in research while doing your clinical practice. I think clinical research is the way forward. Thank you and have a good day. Bye.
So, hello everyone. I'm delighted to address this audience full of doctors. This is, of course, our dream to take whatever we are doing, our research into humans, and of course, we cannot do that without you. So, I'm excited to talk about this today. I've been asked to talk about a little bit about drug delivery as well as the research that we are doing. So, Just to give you a brief overview, in drug delivery, we are taking drugs that are either already in the market or that are newly discovered. And people are discovering a lot of new drugs. Scientists over the period of maybe last couple of decades have synthesized and created a large array of new drugs. These drugs are extremely potent. These drugs are extremely good in terms of what they're supposed to do. However, still a majority of them are not able to go through the different clinical trials or failing at different steps. And I think the major reason for that is an inability to deliver these drugs to the right target, in the right dose, at the right time. And that's basically what we're trying to do. We are more a delivery lab where we're trying to see if we can somehow improve the efficiency of the drug using these pharmacokinetic tools on how to deliver it to a certain organ and not to other organs where it will cause toxicity, to the right amount, not very high or not very low, and of course for the right duration, whether it's for 1 day, whether it's for 1 month, whether it's for 1 year, depending on the application. So this is basically in a nutshell what we do, and we have multiple tools to do that. Mostly we are focused on polymeric and lipid particles. So why particles? These particles are basically nano or micron in size. And what you can do is you can take your drug, you can encapsulate these drugs within these particles, and it does a few things. First, because now the drug has the shell or the surface is covered with a different polymer or a different lipid, it protects it. Let's say when we inject things in your veins, and when we give it through oral route, when we give it through some other injection, and all the serum proteases, different tissue proteases, degrading MMPs, whatever that may act on the drug may not be available that quickly. The other thing it does, it increases the size of the drug a lot. And so, because initially if you look at drug molecule, they are 1 nanometer, 5 nanometer in max. But then with these approaches, you can make them 100 nanometer, 500 nanometer, even larger. And that completely eliminates kidney clearance, because kidney is not able to clear things which are above 10 nanometer or so. So now, a single injection of this will result in a much larger concentration over a much larger duration, because kidney clearance is gone, metabolism is gone with at least a degrading metabolic activity. And so now we can then engineer these lipid and polymeric particles to release the drug at slow pace, and this can be done either through an active mechanism or passive mechanism. Passive mechanism is just the drug is diffusing out from these polymer or lipid vesicles, or the vesicles themselves are degrading in presence of, let's say, water or certain enzyme. And then enzyme can also be site specific. So let's say you want to target cancer and you know some kind of cathepsin is highly upregulated in cancer, you can have these particles that can do that. So that's what we do. There are good examples. There's several examples of this in clinics actually. Doxil, which is a big anti-cancer drug, it's essentially nothing but a doxorubicin encapsulated in a liposome. To keep it short, I think I'll maybe just give you 2 examples of what we're doing in the lab. We primarily work with 2 diseases, osteoarthritis and tuberculosis. In osteoarthritis, and some of this work is published, some of this is ongoing. In osteoarthritis, we are developing carriers that one can directly inject into the joint, in the affected joint, whether knee joint or something else. And these carriers will then release the drug slowly. So for example, let's say if you go to if I have osteoarthritis and I go to clinic and I want painkillers, or I want let's say some other drug that may help improve the function of the cartilage, then the issue is right now if the doctors give you that injection, these drugs will very quickly drain out from the joint through the lymphatics, and you will have really no concentration left after a few hours to a few days. But with these vesicles, we have been able to show in mouse models that you can get very, very sustained release, and that it's very tunable. You can get it for a week, you can get it for a month, you can get it for even 6 months. And that could then open up a lot of drugs that otherwise will be impractical. I mean, you can't expect patient to come in every week and get injections. So with these approaches, hopefully, we can really increase the patient compliance. And just one last quick example I'll give you on tuberculosis. On TB, we are trying to develop carriers, inhalable carriers, that can be loaded with the drug, be delivered through the inhalation route into the lung, which is the primary reservoir of the bacteria. And then, of course, you can now deliver things that are standard TB drugs like rifampicin, isoniazid, or you can deliver things that are new antimicrobial peptides or other small molecules that are being discovered. So these are sort of two approaches that I want to talk about, and I'm happy to answer any questions. If you have, feel free to email me or connect me through some other route. Thank you.
Hello students, this is Dr. Archit Bolur and it's a great privilege for me to address your club, that's the Clinical Cellular Molecular Genetics and Translational Research Club that you are having at the Kilpauk Medical College. I've gone through your activities and it is exceptional that you have done so much of good activities in the last 1 year. On this day, I would like to emphasize upon the need of research in undergraduate curriculum. Remember, it's not about learning what is already known to you, what others have done. It is important that you also do something for the future. So research has to be started, however small, however big it is, does not matter. You will have to take a small topic and possibly research on it and then get into some results which will be of useful to the future. I congratulate the club and I wish the club the best for all their future endeavors. Thank you.
Hello everyone, I am Sujatha and thank you for connecting to me. So this video was a request from the students of the Government Kilpauk Medical College. So the topic that I'll be discussing in this video is network medicine and how it can be used as a support system for the clinicians out there. So the basic idea of network medicine emerged because of the emergence of big data. So what exactly is this big data? So the last 30 years or so has seen tremendous advancement in fundamental sciences, more so in biology. What we have seen is a rise in the generation of data, all kinds of data giving us some kind of molecular information. And since the past 30 years has been focused on generation of data, the current focus is on analysis of this particular data. So where is this data all coming from? That's what the next slide is talking about. The emergence of the big data. As you can see, I have put out the central dogma, which is basically the flow of information from the flow of information from the level of DNA or translating the language of nucleic acids to the language of proteins is nothing but the central dogma. So each of these components or molecular events are associated with the corresponding data types. So with the genetic information or the genomic information, you get the genomic data. Genomics is nothing but an annotation of the ORF or the ORF that I've written out here. ORF is nothing but the open reading frames, which is the part of the genome which encodes for the functional part of functional protein. Now, with that is genomics. You can also extract similar level of information from the RNA or more so for the messenger RNA. That is called transcriptomics. Now, the proteins from the mRNA getting translated into proteins. That information is encoded in the proteomics data. And finally, the proteins are then catalyzing the metabolic transformation, or otherwise called as the biochemical reactions, and provided they are enzyme catalyzed reactions. Now, these metabolic transformations wherein metabolites take part, and all of that information can be extracted from the omics, which is called the metabolomics, which basically gives you the idea of metabolite levels at a defined condition. Now that you have all of this information out there, the next step is for analysis, and use a lot of data analysis tools. One of the methods that I will be discussing today is called the network biology, or the use of networks and models for data analysis. And that is nothing but what is called computational systems biology, which is relatively new field of science, where we integrate all of this information to a level of a network or model. Now, you see all of this information coming in, which is nothing but the different kinds of omics data that you can think. We integrate them all into a model. Let's say we can build models for healthy conditions, we can build models for disease conditions, provided we have the corresponding data available to us. Once the model is built, we then analyze it using a number of model analysis tools that you see here. I've highlighted here in blue. And simultaneously, you can also explore the features of the model, and that few of these techniques I've listed here. Very simplistically put, you can use these model-based methods for high throughput data analysis. That's what we do. We use a lot of these data, publicly available data, omics data, integrate them onto our level to the level of a model. So the crux of this method is integration. And then we get a lot of understanding about the biological processes, be it in the healthy condition or the disease condition. Most of the time, we also highlight the knowledge gaps that are out there. And we are kind of also redefining the biology, because most of the knowledge we are providing is coming from the cutting edge technology and the data that is out there. So now that we've got a fair idea about what exactly is systems approach, that is basically integrating different levels of information and making sense of it, we use it for early diagnosis as well as for designing novel treatments, provided you have got the corresponding data. And now this new treatment or the new drug is actually part of the drug discovery and development platform, which is the next point of discussion in today's video. So we go to the drug discovery and development. And here actually, so there are actually phases on this platform, which actually moves from preclinical research to clinical research. And finally, if it is approved by the FDA, it is then undergoes a lot of monitoring and then getting released into the market. Now, systems biology, network medicine, and mechanistic modeling approaches have been widely used across different levels or different stages of drug development. And this has been used by both big pharma and small biotech companies. And one of the organizations, which is Vantage Research, located in Chennai, that is actually at the forefront of mechanistic approaches that are used to tackle the challenges of drug development. What Vantage exactly does is mechanistic modeling and improves our understanding about the pharmacokinetics and pharmacodynamics as well as the hypothetical mechanism of action of the drug. So simplistically put, anything that you want to understand, what are all the whys that you want to understand between the human and drug interaction, these will help you know about the various drug-related information and via modeling tools. So we use in and out, in silico or computational approaches to understand the drug and human interactions. So that was it about today's discussion and about basically senior scientist at Vantage Research. My research interests lie in human metabolism, understanding the various disease mechanisms and putting the different analysis tools together in identifying novel therapeutics. You can connect to me on my LinkedIn page, and also you can write to me on my email that I've mentioned here. If anything, any term that you do not understand in this video, feel free to write to me, and I'll be okay to make you understand and explain the concepts behind it. So thank you very much, and yeah, look forward to hearing more from you. Thank you.
Dear students, it is my pleasure to speak to you all few words about how you all should be. There are a few points I want to express by experience and as I have seen the world. One is how to connect with the people or public in general and professionally people in specific. What is the meaning of connection? It is a communication. It is not the communication which we all usually do through WhatsApp, online, Instagram and so on. It is a little different from that. It is a connection with the empathy. It is a connection, means communication with the understanding. So what we are doing is totally different. What is exactly what I meant is different. Like in olden days when there are no mobile, no online things, We used to connect, we used to speak to our professors, our colleagues, my friends, teachers, and public in general. So that connection is totally different. It is with empathy, and it is not identified or not found now or going off from the system. So I want to all of you have a better connection with each other, with your teachers in specific as a professionally, and with the public as a social system in general. That is the one thing that is important. You have to have a different approach to learn this one. Second thing is learn to explore new things, new ideas. This our profession requires new ideas. Everything is upcoming technology, so you should be innovative. You should learn new ideas, observe things properly. The system, how to make ourselves is also important. So how to learn, you have to learn. That is a little different. So you should have your own ideas. Another important thing is you should have innovation, you should have interest, you should have involvement, you should have improvise, you should have invention capacity. So these are few I's are important for you to people come up. And fourth one is think new one, have new ideas, and everything and small thing you should explore and learn. Even small thing is very important. You should have a exploration capacity for everything. Last one is but very important. Don't get bad about the failure. Failure is part of our life, part of our profession. Today I do a major surgery, I may lose the patient, but tomorrow another major surgery I may be successful. So you should learn to become both hero and villain in our general life, in society, and also professional life as such. And in profession, this profession is a very dedicated profession, requires undue involvement, attitude, aptitude, learning interest. Otherwise, this profession will become a failure. And all students, I thank again to Sudarshan Bar Association and all students of the Kilpak Medical College and students of the different colleges of Tamil Nadu in specific, and all students from the different colleges of the India as such. I wish all the best. Let all of you become very successful in your life, social life, family life, public life, professionally in specific. Do well, all of you, and thank you very much. God bless you all always.
Hi friends, I'm so happy that my friends from Government Kilpak Medical College have invited me to interact with them. I'm glad that young students under the able guidance of their faculty have floated a club for clinical, molecular, genetics and translational research. It shows how focused you guys are. And if at a young age you know what you propose to do, you will be able to grow very well in the profession. It's good to understand your strengths and recognize them and consolidate them to carry that further, whether you want to go to clinical branches or to diagnostic branches, or you carry out research in later life, or you want to be on the teaching side. I have been in both these sides, both research and diagnostic, as well as on teaching side, and in later years I have been purely into diagnostic and teaching now. But I understand how focused and mature you students are. The younger people have a far greater level of understanding and awareness and will be able to harness those energies very well. You guys have known me through the medium of books, and I'm grateful. In fact, this medium of teaching has brought me closer to a large number of students throughout length and breadth of the country, and I'm so glad that you find this medium of teaching as acceptable medium besides the e-lectures which are available nowadays. Thank you very much and wish you all the best for your club activities, and happy DNA Day. Recently we completed 50 plus 20, that means 70 years of the first description of the DNA model by Watson and Crick, and the entire human genome was released in April of 2003, 20 years back. So you complete 70 years of the initial description of DNA. So wish you a happy DNA and wish you all the best. Thank you very much.
Hi, my name is Bhavani, and today I'll be speaking about the fascinating process of the development of the brain and how defects in this process can lead to neurodevelopmental disorders. Our brain consists of two kinds of cells, the neurons, which are the firing cells, and the glia, which are the support cells in the brain. Our brain consists of 1 billion neurons and 1 trillion glia, and all these diverse cell types are made from scratch during development from a common pool of mother cells called stem cells. Neural development begins when these stem cells divide and differentiate to give rise to the different kinds of neurons and glia. The neurons and glia then wire up and connect with one another to make a functional circuit, much like an electrical circuit at home. This functionally connected circuit then leads to behavior that allows us to seamlessly interact with the world. The process of brain development is controlled by factors which tweak the expression of genes to bring upon programs of making neurons or glia. And the very same process often goes wrong in neurodevelopmental disorders. What are neurodevelopmental disorders? These are disorders in which defects happen in the process of brain development, leading to impaired function. There are broadly two categories: childhood neurodevelopmental disorders, in which the manifestation of the disorder happens during early growing years. Examples of these are autism and intellectual disability. The other is the adolescent or adult psychiatric disorders, in which the age of manifestation is the teenage years. Examples of this include schizophrenia and bipolar disorder. My lab studies the development of a region in the brain called the cerebral cortex in health and disease. The cerebral cortex is the most complex brain structure. It is involved in higher functions such as decision-making, thinking, language, learning, memory, perception of the world, and consciousness. It is also the region most affected in neurodevelopmental disorders. We study the basic conserved mechanisms of the development of the cerebral cortex using the mouse brain as a model system. There are several features of the cortical development that are unique to humans. To study such features and to study patient-specific genetics of the disease, we use something called iPSCs, which are induced pluripotent stem cells, which are derived from the patient cells and are a very valuable disease in a dish human-specific model system. So what we do is take adult skin or blood cells from patients and then convert them into iPSCs using a combination of factors called Yamanaka factors. These iPSCs are similar to the embryonic stem cells present in the growing embryo and thus can give rise to any cell type in the body. We then convert these iPSCs into neurons and glia or into specific 3D structures called cerebral organoids and study which step in neural development is affected in these patients. Such in-depth cellular and molecular studies enable us to understand biological pathways which can be targeted for drug screening and thus pave the way for possible future therapies for neurodevelopmental disorders. Thank you for listening.